Hi everyone, today I'll be talking about Intro to RNA-mediated gene regulation, and specifically I'll be talking about the functions of miRNA and siRNA. So first we have miRNA, which is also called microRNA, and to create miRNA, we will have an miRNA gene within our DNA already, and then that gets transcribed into RNA, which we already know, and then with further processing, that turns into miRNA. With our miRNA molecule, it joins in with other proteins forming something called a RISC complex, which stands for RNA-induced silencing complex. As you guys can see, we have an mRNA right here. This means that this is post-transcriptional, which means that translation will be stopped. One thing you should know about the RISC complex / the miRNA is that it doesn't have to have a perfect base pair matching. This is because each miRNA has several targets, so the mismatch of one or two base pairs won't mean that much. Let's say in our red, we have a RISC complex, and it's going to be complementary bonded to the mRNA via hydrogen bonds. This will create a literal physical block so that the ribosome cannot go through, which will stop translation. Finally, we have siRNA. siRNA stands for small interfering RNA, and essentially it's a double-stranded non-coding RNA, and it's typically around 20 to 25 base pairs long. And for our bodies to create siRNA, we need a viral / transposon RNA introduced, and this is essentially a defense mechanism so we can get rid of it. So our cells will create the siRNA, and with the addition of proteins, will form the RISC complex that we already know of. From there, it'll target the mRNA of the virus or transposon, which essentially degrades the mRNA, preventing translation from occurring. And you should know that this is perfect complementary base pair matching, unlike the miRNA. And this is because we created the siRNA from the direct virus and transposon. Another way to think about this is that we basically steal viral RNA to target the viral RNA. Thank you guys so much for watching, and this was an intro to RNA-mediated gene regulation.